right, welcome back, folks. You can say we are in the middle of the course. Um, Once again, this is a summary video of what you guys need to complete this week and a little bit about what you guys are going to be learning for this week. Week number 8, it goes from today to Saturday, chromosomes and cell division. Um, So make sure once again your book or electronic book um, are good to help you answer questions on the quiz. The lecture notes, I'm going to summarize them. I'm going to go through them real quick. The assessment for this week on the knowledge that you're learning is quiz number 5. Do not worry about this video. I'm still working on it. Not working at this time. So it will not count against you. Don't forget about the virtual lab for this week, mitosis and meiosis, which relates a little bit with what you guys are going to be learning on the lecture portion. Procedures, follow them step by step as you um, answer the questions and enter the data on the data collection and summary worksheet. Completed, uploaded, and submitted, and then take the mitosis and meiosis quiz for the lab. Let's go over the summary of the notes or PowerPoint presentation for the material for this week. So this is actually about DNA. It is time to get to know. DNA is one of the most famous molecules. Lately, we have learned a lot about DNA in the last decade, and nowadays it gets used for a lot of things. You guys know that DNA has been used to convict or to actually crime cases, paternity tests. So we use DNA quite a lot nowadays. So uh, this is actually you need to cover sections on the book, and these notes are 8.1 to 8.8. So immortal cells spell trouble, cell division in sickness and in health. Um, Talks to you about telomeres. Telomeres are actually when DNA replicates the DNA polymerase that puts together the new strand for DNA. This does not add nucleotide to the last template nucleotide, and so it becomes it gives you this uneven, little bit of uneven uh, DNA molecules at the end. So make sure you guys learn about that and how those it affects the health. I mean, it is explained there. Then you will look at the differences in DNA between eukaryotic cells, which is the cells we're made of, and prokaryotic cells, which are the cells bacteria are made of. And so circular DNA versus linear DNA. And DNA in eukaryotic cells is within chromosomes, while in prokaryotic cells is just in the cytoplasm. And so there's a few differences in there. With that, you will learn about the differences in division. Let me actually expand this because it's in cell division between prokaryotic cells and eukaryotic cells. Prokaryotic cells, of course, is a very simple duplication process. That's why they divide so fast. And so it starts with the duplication of DNA. Make sure you guys read and understand how all of that works. Um, so then we go into the, are going to read about the reasons why cells will divide, and then it takes you onto the cell cycle. So cells are always on a stage of a cell cycle, and you can see here basically the cell cycle. take a look at all the phases of the cell cycle. The G1, so there's like a bigger phase which is called the inner phase right here, which subdivides into three phases. The G1 phase is one of them, the G2 phase, and the S phase. And so please make sure that you understand what each one of those phases entitles. Also, then it goes into the mitotic phase, which is actually where the nucleus of the cell divides, and it is allotted to the two daughter cells, which is the end of the cell cycle. Make sure you make a note to understand the G0 phase. Some cells pause in the G1 phase and enter the state called G0, which is a resting phase outside the cell cycle, and they may stay in the G0 phase for days or even years. And some cells, they are always in the G0 phase. Those are nerve cells, and just make sure you guys read about them. Then it goes on cell division. Basically, it just goes of the DNA, how the DNA duplicates. That's a very important concept to understand. So with that, you guys learn a little bit about the structure. I mean, here you will see the structure of the nucleotides within a DNA molecule and the pairing of the bases of the two strands of DNA. And then basically it goes over how DNA, it says a summary of how DNA replicates. Now, in every DNA replication, keep in mind, this process is very quick. There's always errors that do take place, and so there's a proofreading system that correct those errors. I mean, if those errors are not corrected, then that leads to mutations, and so it just tells you in here what I just mentioned. And so it just basically goes on how those mutations can be passed to future generations if they are mating gametes. That is not, I mean, it doesn't talk about that in this particular segment, but we will talk about that next week on but if the mutations happen in cells that stay with the individual, then it talks a little bit about how those that affects. So then it all goes on to basically specifically talking about the process of mitosis. The mitotic phase on the cell cycle is mitosis, and the different stages of mitosis, prophase, metaphase, anaphase, telophase, which is the division of the nucleus and how does that work with the DNA. And finally, when that process is not regulated like it should, so basically you see here it says the details of mitosis, the first stages. Make sure that you guys know what happens to chromosomes, because that's the important concept to be learned, what happens to chromosomes during that process. And then at the end, you guys get when that process is just out of control. It's not working the way it's supposed to work. That leads to cancer cells, and it talks to you a little bit about basically how cells become cancerous or cancer cells because of the process not being of the process of mitosis, basically not of deviations from the cell cycle. I mean, so basically just gives you here comparisons of growing on a petri dish cells, what normal cells will look like versus cancer cells, and so what do they look like, and so how from those cancer cells they lay into tumors, and from tumors that are benign, we can get into malignant tumors, which then they can metastasize. So it goes over all of that process of how normal cells can become cancerous cells. So that is actually the last segment for this week. Um, and so next week we'll continue with meiosis, which is actually specifically the process by which sex cells or gametes are formed. But like always, you guys email me back or request a meeting with me if you guys need more details in any of these concepts. Thank you, and I'll see you next week. Or I'll talk to you next week. Sorry. Bye.